Bacteria.
Bacteria.
Look, there's bacteria. Bacteria.
Bacteria.
You might not see them, but they're there. Bacteria.
Bacteria.
Everything you touch, bacteria.
Bacteria.
That's right, salmonella bacteria. But we have to watch out for bacteria that can spoil our chicken. Bacteria practically everywhere. Everywhere you look, in the kitchen, inside the cooler, in the dining area, in the restaurant.
So my name is John Logsdon, and I'm an associate professor, newly associate professor at University of Iowa, Department of Biology. The name of my blog is Sex, Genes, and Evolution. It basically covers areas that I'm interested in, both scientifically, personally, philosophically. I work on the evolution of sex, work on the evolution and phylogeny of eukaryotes, their relationships, and I'm an evolutionary biologist at heart, so I'm interested in anything having to do with evolution.
I'm Jonathan Badger. I'm a staff scientist at the J. Craig Venter Institute. My blog is T-Taxis. It's named after the genus and species of the American badger. Largely dealing with genomics and evolutionary issues relating to genomics.
My alias and my screen name is Yersinia. My first name is Valerie. I'm from Ottawa, Canada. I achieved my master's in molecular microbiology, and I've gone back to finish a PhD in biochemistry, specifically studying RNA viruses and host pathogen interactions. My username was actually chosen from when I was 15 years old, not to betray my age. At the time we were studying the kingdom Monera in high school and bacteria, and I had to choose a topic for my essay. And I chose bubonic plague, and I was fascinated to know that plague still persisted and that it was such a pervasive illness and that it survived throughout the centuries. And I've come to realize it has a lot of unique virulence determinants that it's shared with others. And so I thought, well, this is a special microorganism and I want to do something to represent that microorganism. I chose the name Yersinia to get people to ask questions about it. And so far, there's very few Yersinias in cyberspace. And I've had a lot of positive feedback about my profession and about the kind of work that I do.
Okay, I'm Elio Schechter and I'm a retired professor. I live in San Diego now, and I teach and write. The name of the blog is Small Things Considered. The blog is about the excitement of microbiology, the diversity of the microbial world, and I pick on sort of somewhat esoteric, unusual, unforeseen, exciting, gee whiz kind of stuff.
My name is Tara Smith. I'm an assistant professor of epidemiology at the University of Iowa. Etiology. Talk a lot about infectious disease, evolution, biology in general, public health, a little bit of politics as it relates to all of those, a little bit of academia. This covers a lot of it.
My name is Larry Moran. I'm a professor at biochemistry here at the University of Toronto. My blog is Sandwalk, named after the pathway behind Charles Darwin's house in Kent. Well, it's very much of a science blog. So I focus on aspects of science like biochemistry and molecular biology. I'm also very interested in evolution, particularly molecular evolution, so I cover that. And of course I cover the controversy between evolution and creationism, which is one of the reasons for starting a blog. We also do the conflict between science and religion.
What is the role of blogging in science? Well, there are sort of two main camps and then lots of things in between. So one camp would just say, people just blogging about what they think about sort of day to day in terms of their science. Another camp wants it to be a little bit more professional, discussion of literature and such. So, but the way I see blogging in science, for me anyway, is a sort of way to get some things off your chest that you might not want to work up in terms of publication, things that are of interest to you socially. So it's a neat way for me to sort of combine science per se along with some of the social aspects that you wouldn't normally do professionally in journal-based format. So I think there's a lot of room for sort of professional science edging in the blogs. That's sort of not why I'm doing it, though. I mean, the main reason why I'm doing it is so that I can have a sort of more informal venue to sort of write about my views, both scientifically, socially, etc.
Well, I think that it'll sort of speed sort of scientific discussions. I mean, I think Although some people have argued maybe the tone needs to change somewhat, I think it's valid that people are beginning to ask questions about maybe sort of the validity of different results, and sort of bypassing the whole traditional way of making a whole paper, which could take months to say if there's a problem with a paper, or things that could be done better that can be done within days or weeks of a paper being published.
It's hard to tell. It's certainly an information transfer mechanism, which I think is useful because it's done on a level which is far more user-friendly than books, lectures, people talking at you, and so forth. It's an easy way to acquire certain knowledge without necessarily going into depth, but having a flavor for certain subjects.
Overwhelmingly, I've had a positive response because in high school, it's not so much looked up on to be scientific, to be the brain, but now I'm coming to realize that a lot of people have appreciation for what microbiologists do and for what scientists and researchers do, and there's a lot of people who are inspired to pursue a career in this field, and I hope that I can be a part of that and convey my enthusiasm for these topics. So overwhelmingly, the response has been positive.
I mean, I get some people on there occasionally who doubt that HIV causes AIDS. Sometimes, some people that deny that evolution happens, some people that deny the entire germ theory of disease. So occasionally there's some friction. Most of the time it's pretty civil and things aren't too controversial, but there occasionally gets to be some heated threads once in a while.
Well, I get all kinds of feedback. I get feedback from people who are very much opposed to what I say, and I get feedback from people who are very much in agreement, and everything in between. And that's the fun of, that's what I find is the fun of blogging, is the discussion and the debate.
What is the greatest microbiological discovery in the past decade? That's really hard. I don't think the thing that has advanced the field, at least my little niche of microbiology, which is infectious disease, has been necessarily a discovery or a breakthrough, but rather kind of a evolution of thinking and going back to kind of the basics of microbial ecology, but having that informed by genomic research, taking all the genomic information we have and applying that to microbial ecology, taking information from culture and from the environment and translating those into a broader picture of infectious disease causation and to microbiology in general. So,
I would say it would be the discovery of using metagenomics, because I mean traditionally bacterial diversity was mostly measured just through some individual marker genes such as 16S, but now that we can get sort of entire sequences of large parts of these genomes, that I think will understand bacterial diversity much more.
Oh my goodness. Well, I see it this way. I think all of biology has undergone an incredible transformation due to genomics, proteomics, all of that. But microbiology on top of that has contributed a tremendous amount to understanding of ecology and evolution. Ecology and evolution are foremost microbial subjects, and I think the awareness, the realization of the diversity and the role of microbes on this planet is unsurpassed.
Bacteria.
Bacteria.
Look, there's bacteria. Bacteria.
Bacteria.
You might not see them, but they're there. Bacteria.
Bacteria.
Everything you touch, bacteria.
Bacteria.
That's right, salmonella bacteria. But we have to watch out for bacteria that can spoil our chicken. Bacteria practically everywhere. Everywhere you look. In the kitchen, inside the cooler, in the dining area, in the restrooms, on our raw chicken. And like I said, bacteria.
Bacteria.
Look, there's bacteria. Bacteria.
Bacteria.
You might not see them, but they're there.